mRNAs are unique molecules, and they've been studied fairly extensively. And as we know right now, everyone seems to know a little bit about mRNAs because of their prevalence in the mRNA vaccines for combating COVID. The cells that I'm currently using are called A549 cells. They are derived from a lung carcinoma sample. We may test a few different cell lines as we go forward. This entire study, this entire field is relatively new in cellular systems. And so we are trying to decipher some of the rules. The first being, what do these RNA molecules that regulate mRNA translation, what do they actually look like? And how can we study them? So that's what my students are working on right now. Currently I have 4 undergraduate students. One of them, Alexandra Fernie, she's been working on a gene called the HIF1A, which is a hypoxia-inducible factor. So it has an mRNA. We have a region that we suspect is playing some kind of a funny business during these stress conditions. And under low oxygen levels, it's triggered to make more copies of this protein.
It feels good. You know, even a little bit of progress in the project is always a big victory. Science is about building knowledge. Our work may help another scientist somewhere else build on their work, and you know, it feels good to contribute to all of that.
Another student that I have is Brittany Benner. She's been looking at a gene called FGF2, fibroblast growth factor 2. Now, growth factor is essential for all cells, right? All cells need to grow, but so do cancer cells. She's been doing a lot of intelligent troubleshooting to get to the point that we can actually rigorously study it.
It's very interesting to be able to work in this lab and do such high-level stuff, because to us, it feels normal at this point. My confidence has really grown. At first, you don't know what anything is, where anything is. And so now I'm to the point where I could come in any time of the day, before class, after class, late at night. It's really my project that I feel like.
I have another student, Jin Young. Essentially, when she came in during the start of the summer in 2022, I was just at the stage of setting up my cell culture facilities. And Jin Young played an instrumental role in setting that up for us. I gave her a lot of independence in reading and researching about best practices to do so, and she has led the effort and taught all of us how to conduct this kind of research in our lab.
I knew I wanted research, so I went on the website. I looked at all the different professors and everything that they were working on for research, and I really liked his. His was like mRNA structures, working with RNA, and then the relation to cancer, which I found super interesting, of course, because I do eventually want to go into medicine and hopefully maybe go into oncology, where this would play a huge role.
They are doing extremely well. Of course, with undergraduate research, you have to understand that they're balancing so many different things at the same time, and they're still growing as individuals. I always try to tell them that, you know, this is interesting to you now. I don't expect this to be your life's goal just because you're working with me. What I want, more importantly, is that you generate that sense of curiosity, that you poke and probe into things and you look and ask why.